Hello students. Today I am going to discuss a very important portion of this chapter known as RNA splicing. Today I will discuss the portion is that is self splicing and alternative splicing. Due to these two processes of RNA modification, the various protein diversity is formed, and from this portion the theory of one gene one enzyme is now abolished, and the new theory comes, that is one gene many proteins. So previous classes we saw that their RNA is modified using the spliceosome machinery, but sometimes without the spliceosome machinery, RNA can be spliced, and this is known as the self splicing. The splicing occurs in all kingdoms or domains of lives, and it is basically very much familiar with the eukaryotic system, but we saw in prokaryotic system that self splicing and tRNA splicing are available. So this is and these splicing of in prokaryotic system, they do not use any spliceosome machinery which is available for the eukaryotic system. So today we will discuss how this self-splicing machinery, self-splicing occurs for the modification of RNA. So there are three classes of RNA splicing. One is nuclear pre-RNA splicing, which is very common and used for most eukaryotic genes. The trans-esterification reactions are happened in the branch point site, and the catalytic machinery is spliceosome. Second one is the group 2 introns, which is rare, and some eukaryotic genes from the organelles and many prokaryotes, they follow the group 2 intron splicing, and it is same as the mechanism is same as the pre-mRNA, and the RNA enzymes that is known as the ribozymes are main catalytic factor this for this splicing. The third one is group 1 intron, which is also rare, and the nuclear RNA genes in eukaryotes and organelle genes and in some prokaryotes this is found, and there is also two transesterification reactions happens, and the branch site is a free guanine residue that I mentioned repeatedly. This is the free guanine residue which is acting as the branch point site in case of group 1 introns. So basically this is the diagram where the three mechanisms of RNA splicings are compared simultaneously, and there is the branch point site is in case of group 1 intron, the branch point is A, and the trans-esterification reactions are very similar to that of pre-mRNA spliceosome, and but the difference is there is no such spliceosome machineries are available, and these catalytic property of RNA is involved in the splicing mechanism. But in case of group 2 introns, there is a free branch point guanine residue which involves the trans-esterification reaction using its 3' hydroxyl group. I mentioned that this is the free, and so that the 3' hydroxyl group is free for this trans-esterification reaction, whereas in case of group 2 intron and the spliceosome machinery, the trans-esterification reaction is occurred with the help of 2' hydroxyl group of the branch point adenosine residue. Next, the working mechanism of group 2 introns and group 1 introns as compared with the spliceosome. In case of spliceosome, the SNPs, those are small ribonuclear proteins, those known as the there are six proteins from U1, U2, U2AF, U4, U5, and U6. These are involved in the spliceosome machinery, and the base pairing between these ribonuclear protein with the pre-mRNA helps the catalytic activity of these proteins, and the splicing happens. But in case of group 2 introns, the self-splicing machinery of the RNA itself, primary RNA itself, is involving in the base pairing of its various domains with the intron, and the branch point esterification was made up, and thus the cleavage of the intron is mediated by this type of catalytic activity. But this catalytic activity of this RNA, now question arises whether it is a enzyme or not. No, this is not an enzyme, because after the working, these introns are degraded. But the main difference is that after any reaction, the enzymes are not degraded, but in case of this, the introns are degraded. So these introns are not enzymes, but these introns have some catalytic activities. In case of group 1 introns, the branch point site is a free guanine residue, and these guanine residues, free 3' hydroxyl group, is responsible for the first transesterification reaction, and in this case, no lariat is formed, and a um, linear intron, 3 introns are removed from the two exons residue, and the base pairing is mediated by the internal uh, catalytic site, or internal guide site, that is mentioned here as the IGS, or blue part this dark blue part is the IGS, which and base pair with the catalytic with the exon intron boundary, and thus the cleavage of the intron is mediated by the self splicing machinery of group 1 intron. So come to the next point, which is alternative splicing. It is basically the step of the protein diversification. In case of T antigen of troponin T antigen, the alternative splicing was first discovered, which contains the 5 exons, and the 5 exons can be joined alternatively, and can form the troponin T antigen and beta troponin T antigen. These two types of T antigens are formed due to the alternative splicing of primary RNA transcript. And this is known as the alternative splicing, where the exon 1 can join with exon 2, and exon 2 can join with exon 3. But in case of alternative splicing, the exon 3 can join with exon 5 instead of exon 4. And in case of beta troponin, these 1 2 exons and 4 5 exons simultaneously join together, and the exon 3 is skipped in this case, and in case of alpha troponin, the exon 4 is skipped. And by this mechanism, from one pre-mRNA gene, the two proteins are we obtain two proteins, which is the result of alternative splicing. So there are there are mainly 5 way how the RNA is modified. One is the normal splicing, and second, these three are not included in your syllabus, so I skip this portion. But for a general information, exon skipping, exon extension, and the intron retention are the 3 more processes by which the RNA can be modified. And the last one is the alternative splicing, by which this is the key step of protein diversification. The RNA alternative splicing is regulated by some activators or some repressor molecules, and this sitting site of the repressor or activator or enhancer binding site is retained in the 3' splice site, which is very much um, required for the RNA splicing. The splice site is also acting as the repressor site, and it is also responsible for the blockage of the spliceosome machinery. When the splicing machinery is sits on the splicing site, the splicing happens, and when the repressor completely bind with this site, this splice site, yeah, then the splicing is stopped, and the primary transcript remain in unspliced condition. But in case of enhancer binding site, this is different than the splice site, which is located somewhere distant in respect to the splice site. And when the activators or enhancers binds to this site, they helps for the binding of the spliceosome machinery, and thus the spliced mRNA splicing occurred. And this is the very important steps of alternative splicing, where the alternative splicing in which the two different exons, those are not contiguous in sequence, contiguous, they can join with each other, and this is mediated by the enhancer binding site, enhancers of the RNA splicing machinery. So there are some inhibitors also I mentioned earlier. Those are known as the RNA repressor. These are heterogeneous ribonuclear proteins, and one of the example is HNRNP1. These binds to the two different splice site. One is 5' splice site, another one is 3' splice site, and the binding of these HNRNP1, this is heterogeneous ribonuclear protein 1, they they helps to they helps by two processes. One is the cooperative binding of more heterogeneous ribonuclear proteins in between these sites, and these binding make the primary transcript unavailable for the spliceosome machinery. Or it can be it can form a lariat like structure, and thus those HNRNP1s interacts with each other, and thus the splicing thus the primary transcript is unavailable for the spliceosome machinery. So students, this is the main three process by which the RNA splicing is regulated. And in the next class, I will discuss the RNA editing, which is another important portion of the RNA modification chapter. Thank you.